Welcome everyone to our AKCI KCA lecture series. We're excited to be joined today by Carmen Priolo from Brigham and Women's and Harvard to talk to us about chromophobe kidney cancer, certainly of interest to this group. We've had some others like Lisa Hensky and others talk about it as sort of an underrepresented but super important disease, at least clinically. So Carmen, we appreciate you joining, and I'll turn the floor over to you.
Thank you so much. It's really exciting to be here. Thank you for inviting me. So hello, everyone. And by way of short introduction, I'm a physician scientist and now full-time laboratory-based investigator. I completed my clinical training in medical oncology in my home country, Italy, and research training at the Dana-Farber Cancer Institute here in Boston before establishing my lab at the Brigham Women's Hospital. So, okay, no conflict of interest. My laboratory studies cancer metabolism, and more specifically, we have an interest in translating biomedical discoveries, biochemical discoveries into therapies and biomarkers that could ultimately be tested in clinical trials. And I will show an example of that at the end of my talk. I really believe that metabolism sits at the intersection of discovery and opportunities for clinical translation. I remember being a student in 2000 and avidly reading the manuscript from Doug Hanahan and Bob Weinberg on defining the 6 hallmarks of cancer. And it is amazing to me how metabolic rewiring it evolved over the 3 versions of the hallmark of cancer. From being supporting features of cancer biology into an actionable hallmark that can be targeted and is really important and relevant not only to the cancer cells, but also to the cancer microenvironment, the tumor microenvironment. So my lab has a special interest in chromophobe renal cell carcinoma. As you all know, this is the 3rd most common subtype of RCC and the 2nd most common non-clear cell RCC type. It can also occur in 3 genetic syndromes, in Birt-Hogg-Diabet and tuberous sclerosis complex, and in Cowden syndrome. In terms of prognosis, patients with chromophobe RCC do usually well, and they undergo surgery to remove the primary mass. However, if they metastasize, then the outcomes are usually poor. Metastatic chromophobe RCC is often resistant to standard therapies used for clear cell RCC. And patients with sarcomatoid differentiation usually have a poorer prognosis, even though they tend to respond better to immunotherapy, and more recent trials validate as initial findings. In terms of a median overall survival, it is estimated to be about 24 months, so even lower than clear cell RCC. And TOR pathway mutations associated with poorer disease-specific survival have been reported in a couple of publications. In terms of macroscopic appearance, these tumors are very characteristic. They look brownish, and these reflect their incredible abundance in mitochondria. A big turning point in chromophobe research came from the TCGA in 2014. 66 tumors and 25 normal kidney samples were sequenced, and the key findings were a consistent chromosomal aneuploidy pattern over focal copy number alterations. A very low mutation rate, only p53 and p10 mutations reached statistical significance in the study, 30% and 9% of cases respectively. And about 23% of cases harbor mutations in the mTOR pathway, including AKT and the TSC genes. Interestingly, 18% of these tumors harbor mutations in genes encoding complex 1 of the mitochondrial electron transport chain, including ND5 and other NADH dehydrogenase enzymes, with very high heteroplasmy, higher than 70%, which is pretty impressive. In our small study of 10 chromophobe RCC, we see the same whole chromosomal losses. And the most amazing thing is that these chromosomal losses always occur and involve the same chromosomes in all tumors. And the chromosome 1, 2, 6, 10, 13, 17, and 21. You can clearly see from this graph. And I should mention that these studies and most of the studies I'm presenting really are in collaboration with Lisansky. We have a very close collaboration here at the Brigham. We really enjoy working on chromophobe renal cell carcinoma together. So interestingly, imbalanced chromosome duplications involving more than 3 chromosomes was considered and reported as a high-risk feature for metastatic progression. That was reported by James Shea in 2017. And I feel like this is very interesting. I'll tell you why in a second. So Chromophobe RCC arises, is believed to arise from intercalated cells of the distal nephron. And now there are several reports that validate this hypothesis and phenotype that has been found. So intercalated cells are critical for the acid-base balance in the kidney. That basically regulate the pH of urine. They secrete hydrogen ions in the urine. And these cells are very rich in ATPase dependent proton pumps. And this is probably the reason why they are rich in mitochondria. And this is why probably chromophobe RCC are rich in mitochondria. However, this mitochondria really become abnormal in the tumors. And you can see here a very old electron microscopy image. I really love this because it really shows that there is so many mitochondria all together here around the nucleus, and then these mitochondria really look abnormal. They look different than the mitochondria in oncocytomas. Those in oncocytomas are more like are swollen and more regular in shape. These have lose their other cristae, or they have very regular cristae or cribriform structures, and they have these vacuoles released by the mitochondria themselves. So it's very interesting. So on the transcriptional levels, the TCGA and other studies mining TCGA data look at expression of mitochondrial DNA genes. And the chromophobe, you can see in the graph here, chromophobe RCC have the highest expression level for 13 mitochondrial DNA genes across all tumors in the TCGA. This is pretty amazing. Something related, a very recent paper from Vincent Zhu and in collaboration with Lisansky showing that mitochondrial plasma proteomic signature in metastatic chromophobe RCC, suggesting that these cancer cells probably shed mitochondrial component or disrupted mitochondria in circulation, into the circulation. So going back to our cohort of tumors, we're really interested in metabolism, as I said, and the first study that we performed was really combining the genomic and metabolomics of this cohort of tumors that we obtained from Chin-Li Vu and Mass General Hospital. We're really, really grateful to Chin-Li for these samples, which were perfectly stored, flash frozen, so we could really do a good mass spectrometry on them. So these are all the metabolites were significant here in the heat map on the left. This is a hierarchical clustering, unsupervised clustering, and you can see as the tumors in green really cluster tightly together, which I think is pretty hard to find. I have done profiles, metabolomic profile in the past of prostate cancers and other tumors, and I think this is the first time I see such a very tight profile. So what was striking to us was that 3 pathways seem to be the most altered, 3 metabolic pathways the most altered in the tumors compared to the normals. The top one was the glutathione metabolism. So all the red metabolites, intermediates here, belong to are components of glutathione metabolism. And you can see how glutathione themselves, both oxidized GSSG and GSH, reduced glutathione, are significantly, strikingly increased compared to normal kidney. These are all paired normal kidney tissues. Despite this increase in glutathione, then we still see a dramatic decrease in the intermediates of the glutathione salvage pathway, which is this here on top, gamma glutamyl amino acids, which was very puzzling to us, and I'll come back to this. So the second pathway that we found really altered was glucose pathways related to glucose metabolism, mostly intermediates of glycolysis, and the pentose phosphate pathways were really upregulated. These are very highly glycolytic tumors. And the third one was a dramatic decrease in intermediates of the TCA cycle, which is not surprising because these tumors are very rich in mitochondria. However, these mitochondria look abnormal, so they are likely dysfunctional, and our finding, I think, support on these hypotheses. So now going back to the most interesting pathway, which is the glutathione. In collaboration with Ed Reznick, we compare our Chromophobe RCC cohort with an additional cohort of oncocytomas that we analyze, again, samples obtained from Chinlivu from Mass General, with the cohort of clear cell RCC that was published by Ariakimi and Ed Reznick in 2016. And the principal component analysis revealed a clear separation of the clear cell RCC from the chromophobe and the oncocytomas, which look more similar together. And that's and that's interesting, but also something that we could expect because two tumors are of the same cell, share the same cell of origin, and share many pathological features as well. When we look at the unique metabolic features of the chromophobe compared to the clear cell, we see that this decrease in the intermediate of glutathione salvage pathway, the gamma glutamyl amino acids, are unique to chromophobe RCC, while both chromophobe and clear cell RCC increase levels of glutathione. Even though chromophobe have extremely high levels of glutathione, which are significantly higher than the ones found in clear cell. So, what is this this glutathione salvage pathway? So, the key rate limiting enzymes of the pathway is a GGT1. These enzymes here, you are all familiar with it. Is a gamma glutamyl transferase 1, is the transmembrane enzymes that is used in the clinics to evaluate liver function, for example. This enzyme catalyzes the degradation of extracellular glutathione into the three components. Glutathione is a tripeptide, is formed by glutamate, cysteine, and glycine. So the gamma glutamate transferase 1 transferred the glutamate residue into an amino acid that is found in the extracellular space, and in forming gamma glutamyl amino acids, which are important into the cell and are used to recycle exogenous glutathione. So what happened in our chromophobe RCC is that this enzyme is basically not expressed in the tumors at all. But the glutathione levels are really high. And so then we looked into the alternative pathways, de novo glutathione synthesis, which is mediated by this cysteine glutamate antiporter SLC7A11 or XCT that it's critical to provide cysteine, which is a dimer of cysteine and is reduced using an NADPH into 2 moieties of cysteine for glutathione synthesis.
Carmen, I have a question.
Yes.
So have you validated any of these transcriptional profiles using IHC on the actual tumor samples?
We haven't, but it's something that we wanted to do. It's hard to identify antibodies that will work well. So I think what I can tell is that there are very old reports showing that GGT1 is downregulated in a few cases of chromophobe RCC. There are very, very old reports from maybe 30 or 40 years ago. That these are the ones that we could find.
So I think it would be really helpful because for two reasons. One is the transcriptional data almost never lines up with the protein data perfectly. And so it would be helpful to know whether these trends hold up at the protein expression level. That's part one. Part two is even the protein expression level does not tell the whole story oftentimes, right? Especially for these cell surface proteins, when you have the transformation event occur, you lose cellular polarity, and now the proteins get completely meshed up in their cell surface localization. And so that can impact their actual functional output quite significantly. So I think it would be really helpful for you to have a sense of what is the functional outcome of all of these proteins in the pathway before you can start drawing up some interesting metabolic ideas over here. I don't know if you've seen Isaac Harris's like recent Nature story on the GGT1 and the extracellular glutathione consumption. Basically, the whole idea is that uh, unlike what we have always thought, their argument is that intracellular glutathione uh, doesn't really matter in terms of redox or whatever else we want to think of. Instead, actually, the exogenous glutathione is working as a reservoir for amino acids for the tumor cells to grow and thrive and survive. So I think it will be helpful for you to know what is the level of activity in this pathway to start addressing it from a completely different perspective. I think biological.
That's a very, very interesting point. So we have done some of that. I mean, we now like switch more on to SLC711 as a main focus of interest, but we have done several experiments. We label glutathione and trying to trace in the amino acids from the extracellular space, doing a genetic modulation of GGT1. So that we have done quite a bit of work in that space. But I agree, that's a very interesting point. And I really believe that, GGT1 is really important for, to scavenge these amino acids in the extracellular space and, bringing them into the cell. So I think it is a very interesting topic. And actually in clear cell RCC, GGT1 is overexpressed. So there is a couple of papers, one from Celeste Simon, showing that GGT1 is actually oncogenic in clear cell RCC. So it is a very interesting pathway. I agree. So going back to the SLC7A11. So we try to validate this finding. This is part of the validation that we have done using some short-term cultures that we have developed in the lab, as well as a line that we were able to obtain from Germany. This was a line generated in 1995. We collected our own samples in this line. We performed SNP arrays and karyotype. And these lines, the Chromo A and our short-term culture maintain the whole chromosomal losses consistent with the patient's Chromophobe RCC samples. And here you see the losses are actually in red instead not in blue. So it's chromosome 1, 2, 6, 10, 12, 13. So most of the chromosomal losses that we find in a human tumor, same thing for the chromo 1039, our line. And then we look at expression of GGT1 in this line compared to normal kidney cells, HK2 or a HAC293. Or a short-term culture isolated from a normal kidney. And we see that in the tumor cells, levels of GGT1 are basically 0. so that the gene is not expressed. Another model that we have incorporated into the studies, and this is really important because this is the only model, preclinical models that actually grows in vivo, are the UK276 cells. So these were isolated in the Marcel Linehan lab from a Chromophobe RCC with sarcomatoid features. These cells harbor P53 hotspot mutation in homozygosity, and the spectral karyotyping show this hyperdiploid karyotype, copy neutral loss of heterozygosity. And going back to what I said in the beginning, so this is considered one of the sort of high risk factors for aggressive Chromophobe RCC. And it is interesting that all the lines, so even these lines that we have obtained from Germany, present the same phenotype. It seems that this is like almost a requirement. It's like a chromosomal endoreduplication seems to be a requirement for the cells to grow in vitro. So at this point, we really focus on SLC7A11, and we asked the question whether these could represent the metabolic dependencies in Chromophobe RCC, as this is a rate-limiting enzyme for glutathione synthesis, and these cells appear not to be able to utilize a glutathione salvage pathway. And inhibition, both genetic and pharmacologic inhibition of SLC711 by either shRNA or the specific inhibitors IKE led to suppression of UK276 cell growth in vivo in nude mice, which was really interesting to us and really suggested that this could be an important target for Chromophobe RCC. If we go back to our, a comparison with other lines, such as the clear cell RCC 7A60 and the Chromo-A, then we see how the Chromophobe RCC derived lines are significantly more sensitive to SLC7A11 inhibition compared to the clear cell. And the clear cell is actually a pretty sensitive model for SLC7A11 inhibition. So this was surprising to us and to many investigators we have talked to. It seems that Chromophobe RCC is really a very hypersensitive tumor to inhibition of this antiporter. And functionally, we can see that when we use the IK inhibitor, levels of GSH dramatically drop in the cells. So this led to a further question. Is this all related to oxidative stress? And Long did an interesting experiment. He treated the UK276 cells with an oxidizing agent. And then these led to dramatic suppression of cell proliferation. And then he used inhibitors of apoptosis, necroptosis, or ferroptosis, trying to rescue these cells. And only the ferroptosis inhibitors, ferrostatin 1, was able to rescue UK276 cells from TPH-induced suppression of cell viability. So now, what is the link between the cysteine availability, glutathione, and oxidative stress? And you all know that I'm talking about ferroptosis. So cysteine availability is critical for glutathione synthesis. Glutathione is critical to regulate lipid peroxidation through a family of enzymes, glutathione peroxidase. You can see here in the cartoon a GPX4, which oxidize glutathione in order to resolve lipid peroxides. So lipid peroxidation induces cell death via ferroptosis. What happens is that these lipid peroxides can be generated enzymatically through the Fenton reaction or non-enzymatically through oxidation of PUFA, polyunsaturated fatty acids specifically, disrupting membrane integrity and promoting cell death. This is where ferroptosis is an iron-dependent form of cell death, mostly based on the Fenton reaction and also as a cofactor of the enzymes that regulate lipid peroxidation. And it was first described in 2012, so it is pretty young new type of cell death by Scott Dixon and Brent Stockwell. And one thing I would like to highlight is that weight is so important, and that the PUFA, the level of the concentration of PUFA in membrane, cell membranes are really important, because PUFA are the only target of these lipid peroxides. And actually, lipid peroxidation can only occur in this specific methyl group, which is a bis allylic carbon, which is located between two double bonds. That's where the reaction is triggered, and then it might propagate to other lipids, adjacent lipids, in the membrane. So to further validate the role of cystine in the type of cell death here, so what we did next was to starve the cells from cystine, and trying to rescue with the same inhibitors that we have used before of apoptosis, necroptosis, and ferroptosis. So here I'm showing that the cells can grow happily with high levels of cystine, usually 200 micromolar, what is present in the media we use in culture. When the levels drop to 20 micromolar, Chromophobe RCC lines, both the UK276 and the Chromo-A, are not able to survive. So viability is low, down to about 10%. While the clear cell RCC-derived 7860 are still able to proliferate. And this viability is fully rescued by supplementation of ferrostatin 1, which is critical to validate the type of cell death and showing that this is really, this is definitely ferroptosis. And in more ongoing work, now in the lab, we were focusing on quantification and validation of cell death. And Sampurna found that treatment with Erastin, which is the precursor of IKE, was the first SLC7A11 inhibitors identified, induces an Exin-5 positivity in Chromophobe RCC cells. These you can see the concentration of 1.25 macromolar here. There is about 47% of cell death between early cell death and late cell death. And this is fully rescued by ferrostatin 1, which was surprising at the beginning. We're now doing more and more experiments. These are unpublished data, so I'm happy to share them with you and get your feedback. So we're now expanding the work to additional cell lines that we were fortunate to obtain through the Tycho lab. And these cell lines, they are CJ, cell line T1, T2, and M, are actually great, and we are further expanding the studies on those. So now, beyond SLC7A11, so I think at this point we have established that SLC7A11 represents a dependency in Chromophobe RCC. However, ferroptosis is a larger chapter, and there is many upstream and downstream regulators, and there is a lot of work currently being done in my lab and Lisansky lab and other collaborators. So I'll show a little bit of different aspects and different perspectives. So this is a very interesting study, single-cell RNA-seq of Chromophobe RCC cells, led by David Brown and Lisansky and many other outstanding investigators in the field. So it is interesting as in a direct comparison of the chromophobe RCC cells with their cell of origin, so the type A intercalated cell in the distal tubule, the ferroptosis is enriched as one of the top pathway with a few components, several components that are involved in ferroptosis suppression being upregulated. And these are NUPUR1 and components of ferritin. So this really suggests that there's definitely these tumors have, there is a big involvement of ferroptosis in these tumors, and many pathways are involved. Maybe some of these tumors upregulate mechanisms of resistance that would be important to know in order to target, to identify and to design the best type of treatment. In this study from Samer Salem, from Lisa's lab, he identified FSP1, if it's ferroptosis suppressor protein 1, as a mechanism of ferroptosis suppression in the cells. Inhibition of FSP1 did not have any effect in vitro. However, when Samer injected cells with FSP1 knockdown in mice, these tumors did not grow well, and there was a very interesting result suggesting that in vivo this mechanism of resistance to ferroptosis might actually amplify and become more important. So one of the major projects that my lab is currently working on is on the concept that lipid remodeling may play a very important role in determining ferroptosis sensitivity and response to ferroptosis inducers. So this concept that membrane lipids undergo quality control, it's a very interesting one. And here, just this graphics really reminds you how higher PUFA in the membrane lipids it really triggers sensitivity to lipid damage, to exposure to oxidants and ferroptosis. Whereas higher concentration of MUFA monounsaturated fatty acids, actually together with antioxidant, can really prevent damage from oxidative stress and support cell survival. But this is a very dynamic concept. So the cell continuously remodels their membranes to shift from a PUFA to a different PUFA ratio. And so cancer cells that are able to shift this ratio and increase MUFA or increase cholesterol and neutral lipids, which are usually sequestered in lipid droplets, is more protected. So this is something downstream ferroptosis that is really important to regulate the response to any ferroptosis inducer. So we were fortunate to be able to obtain the same samples that we have analyzed in the past for metabolomics from Shinli Wu. And so most of the chromophobe RCC and oncocytomas, and also a new set of clear cell RCC. And we perform lipidomics. So this is ongoing work, and I'm showing a summary picture. Hopefully, and this work will be soon published. So what we found is that the oncocytomas, I will start from the oncocytomas, because it's interesting that as a benign tumors, the oncocytomas actually do not change much compared to normal kidney. If you look at the three big families, classes of lipids, sphingolipids, phospholipids, and neutral lipids, the oncocytomas do not change much. However, chromophobe RCC specifically increase levels of sphingolipids significantly, about double the levels of the amount of sphingolipids. And clear cell RCC increase the neutral lipids, which becomes almost replace the phospholipids and sphingolipids. So this is something that we are pursuing, of course, in detail. It's ongoing work, but we're really focusing on the importance of these different classes and very specific species in the sensitivity to ferroptosis and regulation of cell death in the Chromophobe RCC models. So the take-home message so far is that elevated glutathione levels, despite an aberrant glutathione salvage pathway, are a metabolic hallmark of Chromophobe RCC. And SLC7A11 mediated cystine uptake is a critical metabolic dependencies of Chromophobe RCC. And we believe that Chromophobe RCC cells are hypersensitive to ferroptosis induction, and these could represent a very, very important therapeutic strategy for patients with metastatic Chromophobe RCC. So we have different elements here, and we know that there are factors in these tumors that might protect from ferroptosis. The high levels of glutathione, the increased expression of SLC711, GPX4, other ferroptosis suppressors, for example, the FSP1. But on the other end, there are factors that might increase oxidative stress in the cells. And one of these is the mitochondrial dysfunction that is clearly present in these tumors, and especially in those, in that 18% of tumors harboring mutations in the complex 1 genes, a very high heteroplasmy. In addition to other factors that are critical in cancer progression, such as hypoxia, the high metabolic activity, these tumors have very high glycolytic activity as well, and the environmental stress. So in terms of our ongoing directions, we're trying to understand what's the role of lipid metabolism and other pathways, and particularly the transsulfuration pathway, which is the homocysteine biosynthesis, in the response to ferroptosis inducers in the metastatic setting. So we believe that primary tumors are very sensitive to ferroptosis. Now we want to understand whether metastatic Chromophobe RCC preserve this sensitivity, or there are additional pathways that we can co-target to improve sensitivity. And we're also very interested in identifying ferroptosis-inducing compounds among FDA-approved drugs, as the current compounds that we have, for example, AKCI or GPX4 inhibitors, they cannot be used in, they're not ready for human, for clinical use. And I will switch gear for a couple of minutes. I have 2 more slides, and I want to share another study I'm leading, which is based on a carbon-11 acetate PEG trial in patients with renal masses and tuberous sclerosis. So as I mentioned, a Chromophobe RCC, as you know, can occur in tuberous sclerosis complex. So this is a multi-organ autosomal dominant disorder that is characterized by a combination of neurocognitive impairment, autism, epilepsy, and tumors in the brain, in the lung, and in the kidney. And so the 2 unmet clinical needs in the disease, I feel like are really close to the kidney cancer field, are the need for early biomarkers of response to therapy. And this is mainly rapamycin and mTORC1 inhibitors, rapamycin and rapalogs. And also a non-invasive differentiation of fat-poor angiomyolipomas and renal cell carcinoma. So fat-poor angiomyolipomas often need to be biopsied because they're indistinguishable from, they cannot be distinguished from renal cell carcinoma. And so I have like this interest in the metabolic imaging, and together with collaborator at Mass General Hospital and at Yale University now, we have conducted preclinical testing where we have identified fluoracetate as a biomarker for mTORC1 hyperactive tumors. these tumors show very, very high uptake of 18F fluoroacetate. So after preclinical studies, we were able to initiate imaging clinical trials supported by the Lung Foundation Patient Benefit Award in women with sporadic LAM or TSC LAM and renal masses, either rapamycin naive or on treatment with rapamycin for at least 3 months. So our study is a feasibility study of carbon-11 acetate dynamic PET imaging. Imaging acquisition is conducted for 60 minutes, and serial arterial blood sampling is acquired for kinetic modeling.
Carmen?
Yes. Yes.
I don't think it
Sorry,
was a question. Yeah.
just by mistake.
Oh, okay. And so we are completed the study in 6 women, and we are completing the analysis right now. But we had a patient who had in renal cell carcinoma. So we basically will have data on the ability of this modality of imaging and differentiating renal angiomyolipomas and fat-poor angiomyolipomas from a kidney tumor. And I will close here, and I would like to acknowledge the amazing people I'm very fortunate to work with in my lab, current member, and especially Sampurna and Jackie, working on the Chromophobe RCC project, and you've been working on TSC, and Rishi is basically an amazing clinical coordinator who's helped terribly with the imaging trial, and a past member who contributed to the many studies that several studies that we have done, and some of them I've shown here. And Lisanska, of course, my close collaborator here at the Brigham, and the amazing team involved in the imaging trial as well. And I'm happy to take any question.
Thanks, Carmen. Thanks, that was great. I'll open it up for questions. I'll maybe start. So, I mean, as a clinician, somebody searching for targets, I guess my question is, if you were to rank sort of the top therapeutic targets, because when I hear this talk, I hear it sounds like there's a lot of stuff to go after, but we have no chromophone specific therapy, as you well know. We just use our clear cell drugs, and sometimes they work and often they don't. And so I'm just wondering, from your standpoint, is there a top 2, top 3 where you said, boy, if I were therapeutically targeting this disease, I would go after X?
I think that, I mean, if we've done now so much work, I think that SLC7A11, the XCT, is really a big target. That's also really dependent on the enzyme. So there is actually a drug that is an inhibitor of SLC7A11 that is in the clinics. It's sulfasalazine. Sulfasalazine, as you know, is used for rheumatologic diseases, and that is an SLC7A11 inhibitors, which we are trying to test now in the lab and see how we know will perform in Chromophobe RCC. I think from all the experiments, I think this is actually very important, a critical dependency in the tumors, and I hope we'll be able to develop therapies for this target.
And is that the only one on your list? I guess I'm just trying to get a sense, because it seems like we should be able to at least design small pilot studies of some of these drugs, right? And we all have these patients. We give them an IO-TKI or whatever. We give them a TKI, and then there's a lot of these folks sitting around, if you will, waiting for novel therapy.
I know. Well, the other thing is combinations, right? mTOR inhibitors. I mean, the Chromophobe RCC are, with harboring mTOR mutations, they seem to be more aggressive. They seem to be also, the percentage of mutation seems to be higher in metastasis as well. So combining binding mTOR inhibitors. I mean, mTOR inhibitors by themselves, I don't think that would be the solution, right? We all know. So having combination with that would be, I think it would be helpful in a selected population, selected by mutations and other, I think, markers. I mean, I believe like TKI, lenvatinib, I still believe lenvatinib is a strong inhibitor. The point is, going back to the ferroptosis concept, can we trigger ferroptosis using these agents? I think that's also a question mark. It's been shown that mTOR inhibitors, for example, are able to trigger sensitivity to ferroptosis. So more molecular studies, I think, trying to understand more about these drugs will really help identifying patients who could be treated with specific combinations. And in terms of immunotherapy as well, I mean, immunotherapy, the percentage of response to immunotherapy is low, but the latest data show that there is maybe higher rate of response with the Sunny Forecast trial. And that's another interesting topic. Ferroptosis itself could sensitize to immunotherapy. That's why I feel like this is a field that we should try to make more and more effort to identify drugs and maybe FDA drugs that might trigger this pathway, because that could actually synergize with the drugs that are available and they're not very effective right now.
Yeah.
I don't know if I answered your questions. There is a lot of thoughts.
No, there's no
it,
answer really. I'm just curious. I mean, we've been struggling for certainly as long as I've been doing it in Chromophobe, and it's all the responses and data, frankly, are a bit anecdotal.
Yes.
Right.
Yes.
There was 4 out of 9 Len-Ev responses. So we're like, oh, let's use Len-Ev. And then there's Sunny Forecast. I don't remember the number of Chromophobe, but it's all relatively small just because of the rarity of the disease.
I think
so
about 27 with combined.
I'm sure there's signals there, but I'm just not sure how to interpret them, to be honest. So
yes, and I agree perfectly. That's why I think I feel like ferroptosis could be a great target, but we need to really identify the drugs that could be used in the clinics and have and show an efficacy in this molecularly.
Anybody else? Any other questions?
Yeah, just thinking through. I mean, great talk. Just kind of overlaying this. Just thinking through, if we were, because we've thought about this as to Brian's point, right, in terms of therapeutic targeting. We've now had such a surge of antigen-directed therapies in clear cell, and a lot of them don't translate the same in Chromophobe. But there's a couple targets we've thought of, but I think the thing that we struggle with is really how to make sure we're actually testing it. And maybe if you could comment a little bit on models in terms of cell line. You talked about there's really only one really in vivo model, and how maybe sort of focusing on model development can actually help accelerate some of these testings to some degree. But what are your thoughts on the model piece?
Yeah, so that's a great point, and also a painful point, I think, for all of us working on Chromophobe RCC, because we have very few models. So we're trying to generate the short-term culture, and we genomically characterize them to test whether that could be a way to have models that are closer to the parental tumors that could be tested, for example, in drug screenings or other modalities. And that, I think, can be done. But establishing cell lines, and especially cell lines that are oncogenic in vivo, that this has been very unsuccessful so far. It seems that only the sarcomatoid lines can actually grow in vitro and in vivo, and it seems only those with the chromosomal endoreduplication. So it seems like it could be it could be an aspect of those chromophobe RCC that actually progress and metastasize. They need to have that genomic feature in order to progress, and that could be also the genomic feature that is essential for the cells to grow in vitro and in vivo, and for us to like develop more models. So this is my thought. I mean, there have been attempts to generate organoids, really hard with this, with the chromophobe RCC as well. I have experience with PDX, done generated PDX with, with prostate cancer a long time ago. A large cohort. The prostate cancer is very similar, right? Like PDX of prostate cancer do not grow in vivo as well. So I think, that we need to continue to try to generate more cell lines. The short-term culture could be an option. We could think about generating co-culture with those short-term, for example, and maybe trying generating organoids with those short-term culture that somehow maintain the parental tumor genomic characterization. I think you really touched the most sensitive
Yeah, because even if
in the field.
we've thought a little bit about CAR T cell targeting, let's say, but it's like not really clear how to
test, right?
How to test it, even if we create
Well,
it.
so we have done this experiment. We have injected UK276 cells in humanized mice.
Okay.
And we were able to grow tumors. So the beauty of that, and I think that you should actually consider that model, because you can select donors that will have, HLA compatible, it will be HLA compatible with your, with the model, and the tumors will grow, and you will have basically a human system. You have T cells and B cells from the, grafted that will expand in the mouse and will infiltrate the tumors. So that is, we did a feasibility trial. The mice are very expensive, but actually we were able to obtain tumors. And that's something that we want to continue further in order to analyze the tumor immune microenvironment, which will be really important for studies like the ones you are interested on.
Thanks, Ritesh.
I have another question
Any
just
other-
on biology.
Sure.
Just to have your just general sense, like, it seems, when we see these patients in clinic, obviously these are slow growing tumors usually and things like that. But it's always struck me, because, TP53 being, usually an aggressive histology kind of marker. And then it does feel like chromophobe, because of how many very characteristic chromosomal losses, it almost feels like a singular event that pushes it, rather than like you gradually lose chromosomes over time, or you gradually have mutations change over time. It's like, it feels like it's just like that one, but it feels like something that you can imagine with that much chromosomal almost instability, that you could push it to, not be able to tolerate that. Like similar to what you're saying about ferroptosis, right? Not being able to tolerate something protectively. To Brian's point, we have so many different drugs in our armamentarium that are like genomically destabilizing or things like that. It's always kind of struck me that, with something that feels like you've lost like almost a third of your entire genome, it seems like a huge stress on the cell, that it can almost withstand that, and then it grows slowly. Just generally, how do you wrap your mind around just kind of like different biology?
This is why, that's exactly why I feel like the androduplication, so the remaining chromosome will duplicate, and so these tumors actually hyper deployed. They don't have, they can have 60 chromosomes, 49, 60 chromosome or more. So I feel like, maybe we are talking about multi-clonal populations here, and the population of cells that will start duplicating the chromosome is the one that's stronger, the one that will be able to prevail and progress. I was always puzzled by the concept that these whole chromosomal losses actually stay there and the tumors slowly grow with
Right.
half of the genome. I mean,
It feels like on
doesn't,
that-
it doesn't,
Yeah.
yeah, it doesn't feel realistic, right? And I feel like that's a very important point that, you know, was first published by James Shea, and I think it's really important. I think the end duplication, so copy neutral, basically, is a very important point,
like,
and
if
maybe
that
we have
volcano-
underestimated.
So that volcano plot where it's obviously showed all the ferroptosis genes are, you know, so high. On the left-hand side of what's so low is actually all, you know, you basically lose like chromosome 6, right? So you basically lose your HLA class 1. But it almost feels like in that context, you shouldn't actually have total downregulation. Like, I feel like that's why maybe there's a sense of why it's not immune sensitive, right? You have sort of loss of HLA class 1 there on that left-hand side. But to your point then, like if you're losing only, let's say, half of chromosome 6, you really shouldn't downregulate that levels too much, no? Or
Yes, I mean, it's a great question. It's an hard answer, right? I mean, I think we don't know enough of these tumors. I mean, I think there is a lot more genomically than we need to understand. Maybe that's an important point, really, to understand how they grow and why they do not respond to therapies, pretty much to, you know, none of the therapies. So
thanks.
Carmen, thanks for this really good, really interesting topic for the group. We appreciate your time and expertise. So
Thank you.
thanks for coming. Thanks, everyone. See many of you at ASCO or some of you at ASCO in a few days. And we'll see you next time. Thanks again, Carmen.
thank
Appreciate
you.
it.
Thank you so much. Thank you so much,
Sure.
everyone.
Bye-bye.
Bye.